on Friday. Okay, I was telling you about the biological control of the weeds. So I think I have completed that is the terrestrial weeds, where I have covered, that is the 5 weeds. Okay, so one most, I think so it is the open shear I have covered. So where you have the vulgaris and also you have the stricta, and they are mainly controlled by using the cochineal insect belonging to the family Dactylopidae. Okay, so that is the Dactylopius species. Okay, Silonicus we are using one, and other we are using the open shear. Okay, so that is in the central and the south area I have discussed. So coming to the second weed, what I covered is the lantana. Okay, so which has been introduced as an ornamental plant to the Calcutta Botanical Garden. Now it has become a weed, but now also we are breeding the new colored flowers of the lantana, even though if it is a weed, because of its beautiful flowers. And it is mainly, as I told you, it is a seed fly, Ophiumia lantanae, so that is not possible to control. The second one I had told you about the Teleonomia scrupulosa. Okay, so where the main negative back of that weed killer is that, so during the winter, we will be having a severe mortality, and we have a egg parasitoid, so which are avoiding the establishment on the lantana. The third one I told you is the Parthenia. Okay, Hysterophorus, mainly it is controlled by the Zygogramma bicolorata. So the nature of feeding, it is a defoliator, I have told you. And the next I have told you is the Crofton weed, so which is mainly controlled by a defoliator. Okay, so I think so that is, what is the, okay, that is the Parachaetus pseudoinsulatum, so which is a defoliator coming under the family Artidae. So because of the heavy larval parasitation, we are not able to control the weed. I told you about the gall fly, okay, Cecidocharis connexa, so which is also giving, but is not sufficient for the control of the weed because of the multiplication rate of the weed is very high. Okay, another I have covered is the Siam weed, okay, which is again controlled by using a gall fly, that is a Prosidocharis utilis, okay, so which also causes the gall. So these are the 5 terrestrial weeds I have just covered you. Okay, next moving on to the aquatic weed. Okay, so this one I think so everyone has seen. Okay, so when you are just coming towards the GKVK from Majestic, so we have a Hebbal lake. So there we have the plenty of this weed, which we call it as a water hyacinth. Okay, it is Icornia crassipes is the scientific name, and belonging to the family Pododentia. Okay, so this is the family of the water hyacinth. So what is this? Again, if you look into this weed, you can look here. So this is a beautiful free-floating weed. Actually, it is an aquatic plant. Okay, it is a free-floating. Again, it was introduced to India as a ornamental. So in the houses, they used to keep this indoor. Okay, so in the water, you can just keep it, and it is having a beautiful flowers of the pink and the purple color. So that was the purpose why it was introduced to India. But what happened? It has escaped and it has become a problematic, especially in everywhere, wherever we have the ponds, lakes, everywhere. So it is becoming a very problematic weed because of the high multiplication rate. Okay. That is what you can just see here. So it is an origin of the South America, and in almost all the weeds, whatever you take in the major country, other countries also, it comes along the top 10 weeds that have been listed. Okay, so that is the severity. It is not only in India, in other parts of the world also, it is coming under the top 10 weeds which have been listed in their respective country. Okay, so you can see here in the 50 countries, it is a major aquatic weed. And as I told you, it has been introduced to India as an ornamental plant in the year 1896, again to the West Bengal. So here, as I told you, so later it has escaped the whatever you have kept as an ornamental, but it has taken the freshwater, ponds, lake, river, everywhere, whatever we have the water. So it has taken the upper hand in all the water bodies. Okay. 2 lakh hectares of the water bodies has been infected by this. Okay, we are spending lakhs together amount mainly for the control of this weed, especially in and around Bangalore. So what is the problem? Why we are going to control this weed? So as you know that whenever there is a growth on the surface of the water, all the equilibrium, whatever it is there in the aquatic system, it will be damaged. So whatever, especially the fishes, okay, they are not getting the direct sunlight, and that is how, and also these produces the certain chemicals which spoil the ecosystem of the water. So that is the why it is also a major problematic in the aquatic ecosystem. And here you can see as it is affecting the rivers also. Okay, we are exploiting the water for the production of the hydroelectricity. So it is mainly the water will fall from the top, that everyone you know. So it interferes with the production of the hydroelectricity because the flow of the water will be affected. And here you can see the free flow of the water when you are using the water for the irrigation. Okay, in the channel, if it is there, again, the water will not be flowing around. You can see around 40 to 50, 95% of the flow of the water will be affected because of this weight. And fishing and the fish culture, you cannot take when you have this weight. Navigation vessels, it is also affected. Main problem you can see here, evapotranspiration. So as it is also a plant, it gives out the water. So that is a 1.26 to 9.84% of the water is lost as a evapotranspiration. And another is the main thing we have is the breeding of the mosquitoes. You know that why the mosquitoes are problematic. So they transmit the deadly diseases. Okay, whatever we have. Okay, so that is why, because of all problems will be there when we have the water hyacinth when it is growing on in the aquatic ecosystem. So how it has been controlled? Okay, so here initially in the year when it became a weed, okay, so here we went with the identifying what is its native. As we know the native, so that is in the from USA we went with the importation of the 2 weed killers, okay, Neochitina brucki and the Neochitina eichhornia. So they belong to the family Curculinidae. So that includes the weevils. You know that under the Coleoptera, the largest family we have is the Curculinidae. Okay, so when it is a very small weevil, okay, which will be feeding on the weed leaf and also in the petiole. Okay, so that belongs to the order Coleoptera. So these 2 weevils have been imported to India. So it is from the Argentina, that is in the year 1982, we went with the importation. Again, this is the work that is carried by the NBIR Bangalore. Okay, along with this, we also went with the importation of the mite species. You know, mite is not an insect, it is an arachnida. Okay, so Arthogalumna terebranches. So it is a mite, so which is feeding on the water hyacinth. So it is native to South America. So the it is the order is Acari, that is the Arthogalumnidae is the family of the Arthogalumna terebranches. So 2 insects, one mite species have been imported to India mainly for the control of the water hyacinth. Okay, so it is from the USA we have brought it, that is in the year 1982. I will just show you how the weevils. So these are the weevils of the 2 species of the Neochitina species. So these 2, they coexist together. There is no competition, there is no elimination. They are friendly in the nature. And water hyacinth, whenever you just collect, you will find both the species of the weevils on the water hyacinth. So one is a brucki and another is a eichhornia. So how you differentiate between the Neochitina brucki and the Neochitina eichhornia? So one standard character, taxonomic character we are using is here you can see the ridges are there. This is the abdomen part and this is the elytra. And in the weevils, you know that the elytra is fused. It cannot open. So just like other beetles. So elytra is fused in case of the weevils. So on the elytra, so there are 2 sutural ridges are there in the center. So just a midline in the abdomen. So here you can see in case of brucki, so these sutural ridges just below the midline. Okay. Whereas in case of the eichhornia, so here you can see they are just above the midline of the whatever you draw in the abdomen. So this is the one differentiating character that you find between the brucki and the eichhornia. So, and also you can see in case of the brucki, so there is a transverse band is there, moon-shaped band is there on the again on the elytra, but this is not, we are not taking as a striking. These sutural ridges are taken as a main character between the, to differentiate the bruchia and the eichhornia. So here also you can see a half moon shaped band, you can see where you can see the blackening at the top and also below the U shape. And whereas here you will not find that U-shaped band on the elytra. So this is the one you can tell for the differentiation of the bruchia and the eichhornia. So then how they feed? Okay, so here what is the feeding nature of these? How they multiply in the water? Because we will usually know that they are terrestrial. But here you can see the eggs, the one where they lay, okay, in the tender petiole. So wherever you have the eggs of the Neochitina species, they lay the single egg, nearby they can lay. Okay, so these are the eggs of the Neochitina species. Okay, so after 5 or 6 days of the incubation period, so the grubs will emerge. So this is what you can see. As we will, it is a apodous, it is not having the legs. So here you can see the, what is the duration of the grub. So nearly for 1 month, that is the 32 days is the longevity of the grub, and which portion of the plant they are feeding. So here you can see the petiole, you know what is the meaning of the petiole, okay? And the crown region. So these are the portions which are fed by the, that is the grub of the Neochitina. Whether it is a bruchia or the eichhornia, the nature of feeding is same, okay? So mainly the grub will be feeding on the petiole and the crown region, mainly for the 30 days. So after 30 days, so they go for the pupation. So how they go for the pupation? So it is into the water. So where they will be attaching. So the, as I shown you in this photograph, you can see here, this is the free floating. So here they attach to the tip of the root. So which is the live root. So they select the root which is active because they need the oxygen and the carbon dioxide for the respiration. So to the tip of the live root, the pupa will be attached. Okay, so that is the point where the pupa will be attached to the water hyacinth. So it is selecting the live root, attaching the pupa. So it is spending in the pupal stage, it is 23 days. So after 23 days, adult will come out. Okay. So here look into the longevity of the adult. So it is a long days they are living. So that is for the 76 days the adults are feeding. So both the grubs and the adults are the feeding on the water hyacinth. So here you can see both the species are there. So this is a Neochitina, a bruchia, and this is a eichhornia. You can clearly differentiate the position of the ridges on the elytra. Okay, so how they feed? You can see here, they make the holes on the leaf and they will be feeding. So if you find the holes on the leaf of the water hyacinth, so it is a symptom that we have the infestation of the Neochitina bruchia and the eichhornia. You can just pull one water hyacinth which is there in the water and you can just collect them. Okay, so this is what the nature of feeding by the grub and the adults. So the grubs will be feeding on the petiole and the crown, whereas the adults will be feeding on the leaves of the water hyacinth. So what happens because of the feeding by the grub and the adult? So this is what you can see, the drying of the water hyacinth will take place. So what you have to do is that we should not leave this dried weed on the surface of the water. If you just leave it, again what happens? Entire biomass will sink at the base of the water. So wherever you have the ponds or the lake, to the base, it will go and settle there because it is very light, deep, because composting, everything will happen. So because of that, so it will go and settle at the bottom. You should not allow it to settle at the bottom. Again, if the seeds are there, they live for a long time, okay? 4 to 5 years, they are able to survive. Again, if they get the water, they are able to germinate. So that is what the capacity of the seed is also. So because of this, when you find this drying, this mat, everything mat should be clean. So usually they carry the small boards and they remove all this dried mat. So after when the weeds will turn to brown color. So this is what you have to do for the biological control of the water hyacinth. Okay, and also you can see here the female will be feeding at a faster rate. That is a 2.8 times is the feeding rate of the when you compare to the male. Okay, so that means the female is a more damaging one. And how they go for the mass multiplication in the initial stage, because the population was not sufficient, so what they did in the whatever they had, the nursery or anything they had, the shade houses, they took such a large container which are lined with the plastic, and they made the water to stand, and then you put the water hyacinth. Within 15 days it will cover all the water bodies, and then you go for the release of the water, that is the weevil, Neochitina. So they will feed and they multiply, and you can easily collect them and release into the water ecosystem. So this is what they did before when they went for the importation from the other country, and then they did it in the mass multiplication in the field house, and then they went for the release. But now they are not doing this one because we have a sufficient population of both the species of the Neochitina in the nature. So you can just collect and you can release into the water bodies wherever it is there. Okay, so you can see the plastic line, which is a portable. Aluminum tanks also you can use for the mass production of the Neochitina. So this is how it was mass multiplied. And also after the mass multiplication, especially as I mentioned, NBR is located in Bangalore. Okay. And also whatever I have listed, all these lakes, I think so you are familiar. Okay. In the news also you'll be seeing. So especially Vartur Lake, Belandur, you will find the fire. Okay. So that is because of the release of the methane gas in the water, we'll be having that. But what happened? So after the mass multiplication in the shade houses, they went for the field release in important weeds, whatever they have in the lake in the Bangalore. Please, I think so someone is unmute. Please mute yourself. So when they went with the Bairamangala, so where they have a lake, Belandur Lake, Vartur Lake, Hebbal Lake, Nagwan, and Agram. All these lakes we are familiar, and this is the area what it is covered by the water bodies. So especially here you can see they got the 95% control of the weed, okay, because of the release of the Neochitina, Eichhornia or the Brookie, okay. Later, because of the success in Bangalore, again the population was supplied to the Hyderabad. So again in the 8-hectare tank, then to the UP, that is the Gorakhpur, near into the lake they have released, again to the UP, that is the Surra Lake, okay, again to the Assam. So different parts of the country. So they supply the culture for the release. Okay, so this is how they have went with the mass multiplication and release into the different parts of the country. And here you can see because of the success rate of this Neochitina Brookie. So in the year 1982, Bangalore City Corporation, you can see they had given 3.5 lakhs, especially for cleaning. It was 1982. So lakhs means during that time it was having a more value. Now we don't have the value for lakhs. Now everyone will talk about only in crores. Okay. So from the Bangalore City Corporation, it has given the 3.5 lakh mainly for clearing the water hyacinth, especially in the Belandur Lake. Okay. What is the tank? Okay. So just by the release of the weevil, so what is the amount they have saved? Is it 11.2 lakh in the Bangalore alone was the saving, because this amount you are going for the manual control, which is not possible. Okay. And also you cannot go for the spraying in the aquatic ecosystem. So only the way that was left for the control of the water hyacinth was by use of the 2 species of the weevils. And also by that you are not going to spend any amount, no labor cost. Only when the bed is dry, you are going to just remove it. Only during that time you are spending the money. Okay. So that is what you can see here. Alone in the Bangalore during 82 to 83, during that period. So they were saying around 11.2 lakhs of the rupees was saved mainly because of these 2 weevils. Okay. So this is the success story. Still it is doing job, but why it is not controlling the water hyacinth? Because the water hyacinth is having a capacity to multiply into 2 times within 15 to 20 days. So because of the high multiplication rate of this water hyacinth, weevil biology, it is a long, I think so I have seen what is the duration. Okay, adult itself is living for the 72 days. Okay, nearly for the 2 months. So life cycle is very long, but the multiplication rate of the weed is very fast. So both are not synchronizing. So because of this, so the weed is now it is not giving the effective control. It is only because of us, we are contaminating the ponds, lakes, and everything. Okay. And another, I had told you about the mite species, that is Arthogolumna terebranches. Okay. Which came to that is Bangalore in the year 1986, but it was not giving a effective control as it is of the weevil. Okay. It gave only the partial control. So around 25,000 adults were released in Agram, that is the Kangeri and the Bairamangala tanks. Okay, but it has taken the more time. Here you can say 6 months time it has taken for the control. Okay, and also in the Kerala also they went for the field release in the year 1990. Because of the slow multiplication rate, and it is not as effective as that of the two weevil species. So this was not encouraged. Only the Neochitina bruci and the Eichhornia were encouraged, which has given the effective control. Okay, so this is about the water hyacinth. Then another we have an aquatic weed. Okay, so that is the water fern. Okay, so this is not common in Bangalore. So this weed is common in, especially in Kerala. Okay, Salvinia molesta. Okay, so this is the name of the weed. So it is very beautiful. It also came to India as an ornamental plant itself. Okay, it is a water fern and it is a Salvinia molesta. So it is native to southeastern Brazil, and also you can see here it has been spread to Asia, Africa, and the Australia. In all these three countries, it is a major aquatic weed. So then how it came to India? So as it is again as an ornamental plant, it came to India. But where as a weed it was noticed. So here you can see in Kerala, okay? So it was noticed in Ole Lake. So it was in the year 1955. I think so it is the same year when the Parthenium also came to India. Okay, so Ole Lake in Kerala, it was first identified in the water bodies. Okay, so here you can see it has taken almost 11 years to attain the pest status. As you know that, so immediately when they come, they do not behave as if they are the pests. Suddenly they take time for the multiplication, and after a few years, they take the pest status. So this is what happened along with this also. So by 1964, it became a major pest, especially in the water ecosystem. So this is what you can see, the beautiful, it is also a free floating aquatic plant. And here you can see, especially it was very much problematic in Kerala. And also you know that Kerala, they have an important crop is the paddy. Okay, so the paddy crop was severely infected or affected for the growth mainly because of this weed. Okay, so Salvinia. So especially you can see there is one place in Kerala that is a Kuttanadu. If anybody is from Kerala in this class, I think so you'll be knowing the place, which is referred as a rice bowl of the Kerala. Okay, so here you can see what is the that is how much the water body has been affected by this Salvinia molesta. So that is the 75,000 acres of the canal was infected by this Salvinia, and 75,000 acres of the paddy field was affected by this weed. So they were not able to grow the paddy economically. So and also you can see here Kakki and the Idukki reservoirs are the important reservoirs. I think so they are also tourist places. Okay, so they were also infected by or affected by this aquatic weed. Okay, and what happened? And because of this only in Kerala, what happened? They stopped cultivation of the paddy. Okay, so nearly for 1 or 2 years. Okay, wherever they have the severity, especially in the Kuttanadu area of the Kerala. So they were not able to grow the paddy. So this is the status it was the status in Kerala. So for that also we went for the biological control by introduction of another weevil, that is the Cytobagus salviniae. Okay, so it is also a weevil Curculionidae belonging to the order Coleoptera. So it was also in the year 1982. So it is native to Brazil, imported from the Australia. Okay, so here you can see it was again, it is the job again importation. It was done by the NBAR Bangalore. So because of that only, they had multiplied in the laboratory, and it was released in the Lily Pond in Bangalore. Okay, so it was in the year 83 to 84. So whenever you go for the importation, only the small population of the insect will be supplied from the other country. So we have to multiply in the large number. So nearly for 1 year, so they have multiplied in the laboratory, and after in the year 83, so they went for the release. So nearly for the 11 months it has taken for the clearing the weed. Okay, so again in the 1983, you can see here the same NBR, it has signed the 600 adults of the this Siltobacus salvini to, that is the Kerala Agriculture University, that is a Thrissur. Okay, so they were made to release into the Kerala, and also they were given the responsibility of the technology of the mass multiplication of the Siltobacus salvini. Okay, out of 600, so they have to multiply. So this is also, we will, you can see here, Siltobacus salvini, Purpurinidae, Coleoptera in 1982. So here also the multiplication and the feeding, everything is same what you have found in case of the Neochitina. So these are the eggs. Okay, this is the stalk where you can see the stalk, and these are the roots. So in between, the way there is a weeds will, that means the roots will be starting their point. So that is the place where they are laying the egg. Okay, and this is the first instar larvae. So here you can see they have only two instar, first and the second instar. So the first instar, it prefers to mainly to feed on the buds and the roots, because it is, so because you can see that it is near the roots. Okay, so the first instar prefers to eat on the buds and the roots, and whereas the second instar, you can see it makes the tunnel in the apical rhizome, and then it will be feeding. So it prefers to feed on the rhizome. So the first and the second instar, mainly they are preferring on the buds, roots, and another is on the rhizome. So what about the adult? So here adults, you can see it is very small. Okay, you can see it is very small, and it is a completely black color. So adults also feed on the leaves of the this that is the salvinia. Okay, so both the grubs and the adults are damaging, and here also you can find the drying of the entire weed. Okay, and here you can see, so this is the biology what you find it for the Siltobacus salvini. So again you can see, especially in the Kerala, as I told you, so it was a major problem. So State Department of Agriculture, that is in the Kerala, and plus the Agriculture University Thrissur, it was a combined work of the State Department and also the Agriculture University. So within the 3 years, they were able to effectively control the weed, because this weed is not there in Bangalore. Okay, and rarely you will find it. It is only problematic only in the Kerala. So here 2000 square kilometer of the land was cleared by this Siltobacus salvini. And again, you can see here in paddy, just by using the weevil, we are spending only 235 rupees per hectare. Okay, so this is the savings. So by the release of the weevil, they were able to save 6.8 millions annually, because the labor cost is very high. Okay, and just by releasing, you have to purchase the weevil and then you have to release it, and that too in the early time, so they were giving free of cost. Okay, so this is the cost they have estimated per hectare by use of the weevils. You will be spending only 235 rupees, okay, per hectare, and this was the savings because of the release of the weevil. And also you can see here around 1,820 adults of the weevils were supplied to Central Biological Control Station at Srinagar, because just like NBR what we have in Bangalore, so there is also substation in Jammu Kashmir, the Orissa, Andhra Pradesh, and also again the part again to the Karnataka and again to the Kerala. That is a vector control research center at the Shelter line. So there it was supplied and it was released and the control was effective. Okay, and this is how you can see. So this was, so just you can compare. So this was the cover of the Salvinia molesta in the pond or this one. So after the release of the weevil, you can see here it is completely clear. So you can, whenever you are taking such photograph, the location should be same. Okay, the view what you take, it should be same. So that is the view you can see. Okay, where this weevil has Cetobacus salvini has controlled the weevil, that is the weed completely. So these are the two aquatic weeds, so which have been completely controlled by the weevils. Okay, one is the water hyacinth, which is mainly controlled by the Neochitina bruchii and the Eichhornia, and another is the Salvinia molesta, which is mainly controlled by the Cetobacus salvinia. Okay, I think so. With this, I'll just complete the biological control of the weeds. Do you have any doubts? Manju, do you have any doubts, please? Because again, I will just move on to the next chapter. Do you have any doubts? ಗವಿ ಸಿದ್ದೇಶ್ ಮಾತಾಡ್ತಾ ಇದ್ದೀರಾ?
ಮ್ಯಾಮ್ ಕೇಳಿಸ್ತಿರ್ಲಿಲ್ಲ ಮ್ಯಾಮ್, ಸಾರಿ ಮ್ಯಾಮ್.
ಏನಾದ್ರೂ ಡೌಟ್ ಇಲ್ಲ, ನಾನು ಅಪ್ಗ್ರೇಡ್ ಮಾಡ್ತಾ ಇದ್ದೆ. ನಿಮ್ಗೆ ಕೊಟ್ಟಿರೋ ಝೂಮ್ ಐಡಿ ಪಾಸ್ವರ್ಡ್ ವರ್ಕ್ ಆಗ್ತಾ ಇಲ್ಲಪ್ಪ ನಮಗೆ. ಸೊ ದಟ್ ಈಸ್ ಅದಕ್ಕೆ ಪ್ರಾಬ್ಲಮ್ ಆಗ್ತಾ ಇದೆ. ಅಪ್ಗ್ರೇಡ್ ಮಾಡ್ಬೇಕಿತ್ತು ಅದಕ್ಕೆ. ಹೇಳಿ, ಏನಾದ್ರೂ ಡೌಟ್ಸ್ ಇದೆನಾ ಬಯಾಲಜಿಕಲ್ ಕಂಟ್ರೋಲ್ ಆಫ್ ವೀಡ್ಸ್ ಅಲ್ಲಿ? Because after coming back again, I'm not going back to these topics. Okay, can I move to next topic?
Yes ma'am.
Okay. And ಎಲ್ಲರೂ ಮಲ್ಕೊಂಡಿದ್ದೀರೇನ್ರಿ ಊಟ ಟೈಮ್ ಅಂತ?
ಇಲ್ಲ ಮ್ಯಾಮ್, ಕೇಳ್ತಿದೀವಿ ಮ್ಯಾಮ್ ಕ್ಲಾಸ್.
ಏನಪ್ಪ ಏನ್ ಕೇಳ್ತಾ ಇದೀರೋ? ನಾವೇನ್ ಹೇಳ್ತಾ ಇದೀವಿ ಅದ್ರಲ್ಲಿ ಅರ್ಥ ಆಗುತ್ತಲ್ವಾ?
ಎಸ್ ಮ್ಯಾಮ್, ಅರ್ಥ ಆಗ್ತಿದೆ.
Okay, then I will move on to the insect pollinators. Okay, I think so I have completed the predators, parasitites, and then I have completed the weed killers. Next, moving on to the insect pollinators. So everyone, you know what the pollinators means. Okay, so if you just go back to the definition of the pollination, everyone, you know that it is the process of transfer of the pollen, okay, from the anthers to the stigmatic surface of the female flower. So if it is within the same flower, you know it is a self-pollination. If it is from one flower to the another flower, you know it is a cross-pollination. Okay, it is a cross-pollination. So that is the one where in case of the self-pollination, Shreya, in case of the self-pollination, okay, you do not need any external agents for the transfer of the pollen. So just by the wind or anything, so just by wind, they can be, the pollen can be transferred from the pollen to the stigmatic surface of the same flower. So we do not need any external agent as when there is a self-pollination. But when you look into the extra, that is a cross-pollination. So that means the pollen, if the pollen of the same flower fall onto the stigmatic surface, it is not compatible. Even though if it is a pollen on the same flower stigmatic surface, there is no compatibility and there is no germination of the pollen. So the pollen has to come from the another flower. So that we call it as a cross-pollination. And for that, they mainly depend on the agents, or the external agents are involved in the pollination for the transfer of the pollen. They are doing it unknowingly, especially if you look into the bees, they are not intentionally going for the pollination. They are going for the collection of the pollen. When they are flying from one flower to the another flower, they're just transferring the pollen. That is the one thing they are doing. Okay. So for the cross pollination, you need the external agents. Okay. So this is what the definition of the pollination, and what is the self-pollination, and what is the cross-pollination. Okay. Next, moving into why the cross-pollination is important. So especially you look into the whatever the food crops you have, it may be fruit crops or vegetable, any crop you take. So nearly 80% of the crops which are there on the earth are pollinated, are cross-pollinated. Only we have around 10 to 15% or maximum 20% of the crops which are self-pollinated. So that means if you don't have, especially the bees, if you are going to miss the bee population, if you spoil the population of the bees, so it is very hard to get the fruits and the vegetables, especially the fruits, okay, they are cross-pollinated. So that is why you can see here, I have just listed some in different category. Fruits, you take almost all the fruits we are eating. The citrus,